Hello, everybody. My name is Fadhl Alakwaa. I am a faculty in the University of Michigan. Today, I will talk about integrating transcriptomics, proteomics, and metabolomics data from the CPROBE cohort to identify mechanisms underlying chronic kidney disease. So data integration is a mathematical approach to integrate multi-dimensional data from different omics data. It offers a good tool to better understanding the biology of the kidney. It has many applications in drug repurposing, drug discovery, and biomarker discovery, and also in disease classification. In the CPROBE cohort, we have a good opportunity to apply to integrate multi-omics data type for two reasons. First reason that for 37 patients, we have four omics data type. So for this 37 patients, we have transcriptomic profile and plasma and urine proteomic profile, and also the metabolome profile for this patient. The second reason that these patients, they have a good follow-up time, at least 5 years in follow-up time, and also a third of these patients, they reach the outcome. When I say the outcome, I mean when the patient, they achieve end-stage renal disease or 40% decline in their eGFR. So here what we did, we applied two integrative approach, MOFA, which is unsupervised approach, and also Diablo, which is supervised approach to integrate these four omics data from these 37 patients. So the first one, we apply MOFA to the CPROBE cohort. And as you can see, when we integrated 6,000 features, we are integrated and combined to form 7 factors. So this 7 factors computed from linear combination of these 4 omics data type. And now the most important question for these factors is which of these factors associated with the disease progression, or which of these factors capture the disease progression. And as you can see from this Kaplan-Meier curve, that one of the MOFA factor, which is factor number 2, was associated with disease progression. And also this factor number 2 also was driven by the urine protein omics data type. When we perform the pathway enrichment analysis for the top features extracted from MOFA factor number 2, complement and coagulation cascade pathway was enriched in at least 3 omics data types. Some of the pathway, they only enriched in unique, some specific omics data type, like for example, purine metabolism only enriched in only the metabolite. So here, MOFA was successfully applied to CPROBE data. One of the integrated factor, factor number 2, was associated with disease progression and driven by urine protein, and complement and coagulation cascade pathways was enriched in at least 3 omics data types. Second, we apply another approach to integrate these 4 omics data to the CPROBE cohort. And when we applied Diablo to this cohort, we identify 108 features associated with disease progression. Again, when we perform pathway enrichment analysis for Diablo identified features, a complement and coagulation cascade pathway was enriched beside the JAK-STAT signaling pathway. So Diablo was successfully applied to CPROBE data. 108 features was identified by Diablo and was associated with disease progression, and also again complement and coagulation cascade pathways was enriched in at least 3 omics data types. Trying to combine the results from the MOFA and Diablo, in this Venn diagram, what you can say that at least 8 urine protein, they shared between MOFA and Diablo, and they form function function networks, as you can see from this protein-protein interaction from string database. And interesting that these MOFA and Diablo urine proteins, they are associated with disease progression, as you can see from the Kaplan-Meier curve, and we plot here C9 as, for example. So in summary, what you can see that we integrated 4 omics data type from CPROBE cohort using 2 different approaches, and MOFA factor numbers was associated with disease progression and mostly captured in the urine proteomics, and 8 urine proteins were shared between MOFA and Diablo. Complement and coagulation cascade pathways enriched in both approaches. Second, we are trying to validate our finding in another independent cohort. For this reason, we have the urine protein or profile for at least 94 independent samples, and they have a 4.5 follow-up time, and 33 of them, they achieve the outcome. Patient in the independent samples, they are older and they have lower eGFR and low UECR. So what we identify, from this first plot, you can see that MOFA and Diablo shared urine protein are shared with disease progression in the validation sample after adjusting for eGFR, age, and UECR. In conclusion, both MOFA and Diablo identify a network of genes of protein enriched for complement and coagulation cascade pathways. A set of urine protein would identified by both approach also consists with complement and coagulation cascade pathways. Multiple urine protein were validated in independent samples, and prior analysis in independent cohort also highlight genes differentiated set of urine protein associated with the outcome in CTB. Last things, I want to thank every member in our data integration working group. And also I want to thank the funding sources. And thank you very much.